Welcome to our lab podcast on the digestive system. We're going to start at the beginning, the superior end. So we're going to start in the oral cavity and look at the histology of the lip as we make our way down through and into the esophagus. So here we're looking at a fairly low magnification view of the lip. And with the lip, we know we're going to have a transition zone, which is going to be in this location. And it's going to be a transition from our surface thin skin type epithelium to our oral mucosa. Now at this low level, again, we see this tissue running through the middle. And you should begin to be able to recognize that as muscle. So that's going to be our orbicularis oris. We can pick up some of our minor salivary glands present on the oral side. Over on the skin side, we can recognize sebaceous glands, hair follicles, stratified squamous keratinized epithelium. Then we get into our nice expanded reedy apparatuses present in the vermilion or the transition zone of the lip. Closer magnification, looking at the skin surface. Again, you would be hard pressed to know if you're looking at just thin skin or a portion of the lip, because here we can see a hair follicle coming in. The hair has been removed. This is just the hair follicle associated with its sebaceous glands. The hair's been removed. Here we can see other sections through that same hair follicle, more sections of sebaceous gland. We have our dermis, our dense irregular connective tissue. So it looks just like thin skin. Now, as we move into our vermilion zone, here we can see the expanded reedy apparatus with these papillae projecting upward into the epidermal layer. We can see our stratified layers, our keratinized layers here, transitioning more to the parakeratinized like we see in the oral cavity. So that's the nice transition. Again, really marked with those papillae. And here we have the sections through our orbicularis oris. Now, on the oral side, again, we can see orbicularis oris sections on the right. We've got our parakeratinized oral type epithelium, but then we have our fairly substantially mucus. We may have some serous in here as well, but primarily mucus in our minor salivary gland that's going to empty into the oral cavity. Now, in this case, as we're looking deeper into those layers, we can see the distinction here between a mucus type minor salivary gland and a serous type salivary gland, much more densely stained. Now we're going to shift and look at the surface of the tongue. And this is a cross section through the tongue. And understand we're mostly looking at muscle as part of the tongue, but we're also going to have some lymphoid type tissue, secretory type tissue present here in some of our glandular material that we'll look at. So in this particular section, what we're seeing, this darkly staining material, this is going to be part of our lingual tonsil. These are our lymph nodules that are present in part of that. We don't necessarily see the crypt organization, so a little bit out of focus, but that's going to be part of our lingual tonsil. Now on the dorsum of the tongue, we're going to begin to look at the different papillae that are present there, as well as the taste buds that will reside on some of these papillae. So in this particular illustration, we're looking at two types. We can see the filiform papillae. We don't see them quite as spiky as we're going to see in other sections, but these are our filiform papillae. Here we can see the different sections through the different orientations of muscle. We have connective tissue. But then in some, we can see these rounded tops to these papillae. We can see the dermal papillae projecting upward. This is going to be our fungiform papillae. And we do have a couple of taste buds, those little barrel-shaped structures that we see right there at the surface. Now in this particular image, we can see on the dorsum of the tongue, mostly filiform papillae, but then on each side that we can see inset on the left, these are going to be our circumvallate papillae. Now again, we're going to get closer and closer in our magnification. Again, filiform papillae here on the right, filiform on the left. This is our large circumvallate papillae. We can see the rounded top in the middle. We can see the deep depressions on either side. These serous-type glands are going to be our glands of von Ebner that are emptying into the groove of our circumvallate papillae. And it's going to be on these lateral surfaces where we will see our taste buds. And you might see those little light-colored spots. I believe those are going to be some of our taste buds we'll look at. Closer and closer magnification, we can see our taste buds here on this lateral surface. We can see our serous glands. Here we see a duct of the von Ebner's glands emptying into the groove. And then we have sections through muscle throughout the remainder. So here is our taste bud. We can see the taste pore. We would have the microvilli of the cells projecting through, detecting the chemicals or the tastants. Those will be transferred to neurons that are present down at the base. Processes of neurons are going to detect those signals and transmit them onto the brain. We can see one in nice focus, portion of a second, portion of a third, and maybe a hint of one out of the plane of section here. These are great examples of our filiform papillae, these spike-like papillae that are sharply keratinized on the surface, and certainly our feline and canine relatives are going to have those very stiff bristle-like papillae on the surface of their tongue. No taste buds are present on the filiform. They're only present on the fungiform and the circumvallates. This is laying down sideways, but we can see the multiple layers of the keratinized surface here of our filiform papillae. Now we're going to begin our way to go down the alimentary canal. So this is the very beginnings of the alimentary canal. This is the esophagus. And we mentioned in the lecture podcast, the esophagus is going to be characterized by the infoldings of the lumen. You can see how collapsed this looks. This is going to be the portion of our mucosal lining. Remember, we have mucosa, and that's going to extend from the epithelium, lamina propria, and then we can see our discontinuous muscularis mucosa. We get into the submucosal region here. Then we have our muscularis externa, which is typically inner circular, outer longitudinal. We can see these going almost around in this direction. You can see these elongated fibers. These are more cut in cross section because this is a cross section view of the esophagus. These are coming straight at you and going away from you, and these are going around in a circle. And that's how we can identify circular versus longitudinal. And then here, likely this is going to be adventitia because that's the bulk of what we're going to see with the esophagus. Now, as we look at higher magnification at these different layers, we've got our epithelial layer, part of the mucosa, lamina propria, muscularis mucosa, submucosa, circular and longitudinally arranged smooth muscle, or skeletal, depending on where we are, remember. And this is going to be part of our muscularis externa. We don't see the adventitia out here. And in this particular slide, looking at the stratified squamous nature of this epithelium, seeing these mucus glands here in the submucosa, this is an esophageal gland. So all of these things together give us the impression that we're looking in the esophagus. Here we see another section of esophagus, a different stain, and what we're seeing with the mucosal layer. We can see the epithelium, lamina propria, discontinuous muscularis mucosa. Here we have the submucosa and the muscularis externa, followed by the adventitia. This is a fairly thick-looking submucosa, We've got some vessels in here, such as we see here. And again, really not collapsed like we're used to seeing. This is sort of atypical for what we see with the esophagus, but we can make out those multiple layers. Once again, examining more closely, we can see the stratified squamous nature of our epithelium, lamina propria, discontinuous smooth muscle of the muscularis mucosa, dense irregular connective tissue of our submucosa, and then we have our muscularis externa. Now here we're approaching the junction between esophagus and the stomach. Now this is sort of a crazy section here because we have this fold that we've tangentially cut through the epithelium. That's really what we're looking at here. And so we can recognize our stratified squamous epithelium here. We've got our muscularis externa down here, this sort of thicker muscular portion. This is going to be part of our gastroesophageal sphincter. But then as we get to this side, we can begin to pick up our gastric pits and gastric glands. So this is going to be stomach, this is going to be esophagus, and this is just that crazy fold where we're sectioning through epithelium, getting a little bit of lamina propria there. Again, here we're looking at that junction between esophageal epithelium and then our gastric mucosa in this area. There we can clearly see our gastric pits, our gastric glands, so we know we're in the stomach. And here's a low magnification of a similar shot. Again, we can see our stratified squamous epithelium, submucosa, muscularis adventitia, possibly getting into serosa, because now that we're down at the level of the stomach, we've passed through the diaphragm. And once we pass through the diaphragm, now we're going to have a mesentery, and that is going to be then called a serosa on this outer border. So that could be what we're seeing right here, that mesentery and the serosal layer.